The second topic of today's lecture is epidemiology. And you probably already know what is the concept of, what is this science? What is the objective of this science, right? This is a science that is going to um, study the distribution and the cause of diseases in the population. And then epidemiologists are like detectives that collect and compile data to look for the cause of that disease, how they are transmitted, how they are distributed in a population, the potential risk factors associated to that disease. Let's now go through some important concepts related to epidemiology. And the first one that I'm showing you here is the concept of incidence versus prevalence. Something that you have to keep in mind is that epidemiologists are they're more concerned with rates of infection rather than the absolute number of cases. And the best way to understand this is to consider small versus large cities and how the rates will be a more important thing to look at than the total number of cases. Prevalence, right here, is the proportion of cases in the population at a given time. On the other hand, incidence is the rate of occurrence of new cases, and that incidence tells you about the risk of contracting the disease. Prevalence indicates how widespread the disease is at a given time. Here is a hypothetical example to understand the difference between the two of them. Think about a disease that takes a long time to cure, let's say many months, several months, and was widespread in 2019, but then new cases started to significantly decrease in 2020. This disease will have both high incidence and high prevalence in 2019. But in 2020, where no new cases are being reported, however, people have not recovered from that disease yet when they move from 2019 to 2020, because it takes several months for that disease to develop and to the person to recover. In 2020, it will have a low incidence, no new cases or few new cases, but because of the long time that takes to cure, the cases that accumulated in 2019 will persist in 2020, and then the disease will still have a high prevalence. Let's take a look at this picture to understand that better. Incidence will be the number of new cases right here. Prevalence are the number of cases that are accumulated over time. Some of them may recover, and then now we don't account for them. Some of them may die due to that infection. And then we keep accounting for the total amount of those cases at a given time here, while the incident will be only the new cases that are showing up at a given time. Now, some other important concepts related to epidemiology: morbidity, mortality, case fatality rate. Morbidity refers to the level of health and well-being. Mortality is related to the risk of death. For example, I was looking at the US estimates in 2017-2018 for the flu, and 45 million people developed illness. And from those, 61,000 died from the disease. So the other thing is here, mortality versus case fatality rate. What is the difference there? You know, in order to understand this better between mortality and case fatality rate, let's take a look at a hypothetical example. Let's do this. We're going to consider two populations. Population one consists of 10,000 individuals in that population. Five hundred have been diagnosed with the disease. And let's say 100 have died. So again, this is the this is the total population right here. Let me use a different color here. 10,000. And this is for population number one. In this population, if I look at the mortality rate for the disease, the mortality rate will be 100, those who died, divided by 10,000, the total population. And then we will multiply that by 100 to get a percentage. And that will give us 1%. Now, if I want to look at the case fatality rate, the case fatality rate only accounts for those who were diagnosed with the disease. So in that case, the rate that I will establish will be 100 divided by those who were diagnosed, 500. Multiply by 100 to give the, to get the percentage. And that will give me a case fatality rate of 20%. So again, this is case fatality rate. And here, this is mortality rate. Now, if I go and look at a different population here, I look at population number 2. In that population, I also have 10,000 total individuals. In this case, I have 50 people that were diagnosed with the disease. And then I have 45 that die. What are going to be the rates here? So the mortality rate will be 45, right? Divided by 10,000, the total population. Multiply by 100. And then now this will give me a 0.4% in terms of mortality rate. However, if I look at the case fatality rate, even though I have less people dying here, but if I do that rate based on the people that have been diagnosed with the disease, that will look to me like, something more scary in terms of, the ability of that pathogen, once you contract the disease, the ability to die from that disease. Multiply by 100, this is going to be 90% of case fatality rate. So hopefully this particular example, show you how these two rates can give you a very different, interpretation of your data. And you have to be careful how you analyze that. Maybe in this case, we are in the presence of a disease that is more easily transmitted. That's why you have more cases. Here, maybe this is a disease that is not transmitted that easy. However, if you contract the disease, there is a good chance that you may die from that disease. Other concepts that you have to take into consideration, these are related to disease distribution. When we talk about endemic versus sporadic, epidemic, and pandemic, in the case of endemic diseases, we are talking about a disease that is regular, that there is a continuing occurrence in that population that is constantly present. A good example of endemic disease is right here, common cold, that is all the time circulating and happening. Common cold that can be produced by rhinoviruses or human coronaviruses. I'm not talking with that common cold, and I'm not talking about the SARS-CoV-2 that is circulating now. These are other human coronaviruses. Sporadic disease will be a disease that has an occasional occurrence. I would say a good example of this would be Yersinia pestis. The bacteria that produces the plague. Today, most human cases are sporadic and occurred in rural areas. However, it was a terrible widespread disease back in Europe in the 13th, 14th century. It's not the case anymore because rats are under control, and you will understand when we discuss the disease, you will understand why rats play an important role here. And also because today we have antibiotics that can be used to treat this infection. There are two different types of plague infection. You have the pneumonic plague that is really high case fatality rate, and you have the bubonic plague that is not that high in that case, and that could be easily, rapidly treated with antibiotics. Then we have the epidemic, diseases. Here we have a significantly increased occurrence. That's what is happening, for example, or what happened back in 2003 with the SARS coronavirus 1, not number 2, the one that is circulating now, that is pandemic. I'm talking about the one that happened back in 2003, the SARS coronavirus 1. Most cases were detected in Asia, in a few countries. Also some cases, for example, were detected in Canada. That resulted in a total of a little bit more than 8,000 cases and a little bit more than 800 deaths. And that was an epidemic. Now, when the pathogen now is spread all around the globe, and now you have a global what we call a pandemic, something that is distributed and is present in almost in every continent. Now we're talking about a pandemic, and that's what is happening currently with the SARS coronavirus 2 pandemic that we are living. Is also what happened with, for example, with HIV. This pandemic is everywhere around the world. So we were talking about vaccines before, and how they can be used to prevent infectious diseases. Another important factor to prevent infectious diseases is to understand the chain of infection that goes through these steps that are outlined here in this particular slide. Let's now go one by one in the following slides, starting with reservoirs, transmissions, and susceptible hosts. First thing here is reservoirs. A disease reservoir or a reservoir of infection is the population of organisms or the specific environments in which an infectious pathogen naturally lives and reproduces. The pathogen depends on these reservoirs for the survival in nature. Some of these reservoirs, they will be infected with the pathogen, but the pathogen is not causing disease in them. And then now they can spread it to other susceptible hosts that could become what we call a terminal host, in which the pathogen can replicate and produce a lethal disease. Sometimes the reservoirs are just environmental, soil and water. The pathogen is present in soil, for example, Clostridium tetani. Or the pathogen is present in water, for example, Vibrio, cholera. And then now from that environment can be transmitted to a susceptible host. There are diseases for which humans are the only reservoir. That's the only organism in which some pathogens can replicate. It was the case of smallpox, the only pathogen that has been eradicated thanks to massive vaccination. This was a terrible disease. It has been written in history books that one of the reasons why many populations in the past where people were dying in high numbers was a lot due to this particular pathogen, to smallpox, in addition to other reasons. But the smallpox was a terrible disease. Terrible disease with a mortality around 30%. So that means a third of the people that contracted the disease could die from this infection. And thanks to vaccination around the world, this disease was eradicated and is not circulating in this planet anymore. The only samples of those viruses are, in high security laboratories where samples are kept, in case other studies have to be done. It's a vaccine that is not given to the population anymore because since the pathogen has been eradicated, there's no need to continue distributing that vaccine in the population. Now, when you have a pathogen that only targets human, and you get a vaccine as effective as this one against smallpox, you have a good chance of eradicating that disease. And for example, we were talking about the measles, mumps, rubella, and varicella zoster vaccine. These are 4 pathogens that only infect humans. If we are able to massively vaccinate all the population, and we could get rid of these 4 viruses from this planet. Polio virus. This will be probably the second human pathogen that could be eradicated thanks to vaccination. We are close to it, but still there are few cases being detected in a few countries. But as soon as we don't detect any more polio thanks to vaccination, we could call that the second disease that will be eradicated. But again, it has not been eradicated yet. The only human disease that has been eradicated thanks to vaccination is the smallpox. Now, if you have a pathogen that lives also in another animal, wild animal, a pathogen that is present in soil, you know, you could have good vaccines that you could give to people to prevent those infections, but those vaccines will not eradicate the pathogen from the planet, because there is a constant source of that pathogen in places where you cannot eliminate them. So you will have to continue vaccination from one generation to another one, you know, in the future. Would be the only way to prevent those infections through vaccination. So this has a little bit more about you know, human reservoirs and animal reservoirs and environmental reservoirs. I actually mentioned, you know, most of those things in the previous slide. So a lot of the things that I mentioned before are here, you know, with the only exception of this case of asymptomatic infection carriers. Remember that there are subjects in which a disease may not show, or the infection by a pathogen may not show signs or symptoms of the infection, and still this person could shed that pathogen to others and could transmit it to others. That is the case of Neisseria gonorrhea. Up to 50% of women infected with gonorrhea are asymptomatic and they can transmit it. Also, Staphylococcus aureus is asymptomatic in many people and they can transmit it to others. And symptomatic infections, well, in that case, it's easier to identify the source of the pathogen and to try to stop it. You have here a lot of different animal reservoirs. And an important concept that you need to know is that a disease in which an animal transmits a pathogen to a human, we call that a zoonotic disease, zoonosis. The disease in animals and now can be transmitted to humans. Generally, you know, these are the pathogens that in the animal maybe are not causing a severe disease, but then humans, when they get exposed to that pathogen, they develop a severe disease that in many cases can kill the host. And that's why we use in these cases the term of terminal host. And then finally, you have the environmental reservoirs that I discussed in the previous slide, talking about pathogens that are present in soil, in water, and then from there they can be transmitted to humans. In terms of portals of exit and portals of entry of pathogens, well, depends on the disease and how, you know, what is the route that the pathogen used to enter our body and what is the way that is getting out of our body to be transmitted to another subject. It could be through a body surface or orifice, you know, that could be the entry, exit route for the pathogen. Intestinal tract, you know, oral fecal transmission in this case, respiratory tract, respiratory infections, droplets that could be created, you know, in droplets of saliva when we talk, when we sing, when we sneeze, when we cough. And then these droplets of saliva or mucus can contain those pathogens like tuberculosis or many respiratory viruses. The skin could be the surface of choice for some pathogens, Staphylococcus aureus, and then when the skin starts to shed skin cells, then now that bacteria that is there could be transmitted to another person. And then you have those pathogens that are sexually transmitted. The pathogen could be present in the semen, in vaginal secretions. It could be, you know, also related to sexually transmitted. It could be also, you know, other mucosal surface in the body, like oral or the anal mucosa that could be also susceptible to these pathogens. In terms of the transmission of pathogens, well, there are different ways in which transmission can happen. The initial transmission of the pathogen to the host, you know, it could be through direct contact. These are usually pathogens that are not able to survive outside of the host for too long, and they require that close contact for a more effective transmission. It could be vector-borne. Many of these cases are highly virulent in human hosts. And another thing that you have to keep in mind is that sometimes some of them may have a greater ability to survive outside of the host, and they become more virulent and easier to be transmitted. Let's now go through the different transmission of infectious diseases. You have here different ways in which diseases can be transmitted. To the right, direct transmission. To the left, indirect transmission through vehicles. Let's go through each of them. So horizontal contact, the first example here for direct transmission. These are pathogens that require that close contact, kissing, sexual intercourse, that are different pathogens in this particular group. For example, HIV is a case. HPV will be there. Chlamydia trachomatis. Trichomonas vaginalis. We will talk about all of these in sexually transmitted infections. In this group, we also have droplets as a potential way for the transmission. When we talk about droplets, just droplets, not droplet nuclei, I'm talking about droplets here. We're talking about big droplets that are produced. These are big droplets of saliva, mucus, that do not travel that far away. So in order for the pathogen to be transmitted, it requires a close proximity. And a typical example is the SARS coronavirus 2. That rule of the 6 feet separation in order to prevent these droplets from reaching, another susceptible host. In this group, you also have, influenza, although influenza also could be transmitted through droplet nuclei, right? But you also have all the viruses that produce common cold. Those are all pathogens that are transmitted through these droplets mechanism. That's the main way of transmission. Then you also have pathogens that could be transmitted from the mother to the fetus or the newborn. In both cases will be considered vertical transmission right here. And typical examples of vertical transmitted infections, well, the rubella virus, the hepatitis B virus, the varicella zoster virus. Those are all vertically transmitted, or can be vertically transmitted. They can be transmitted through other routes too, for example, through droplets. They could be transmitted. But these are typical examples of vertical transmission. Now, indirect transmission, I'm giving you different possibilities here. For example, fomites is the first case for indirect transmission. Fomites means that, for example, I cough and I cover my mouth with my hands, and then now with that hand that maybe contains a pathogen, I will touch a doorknob like you see there. Then I'm placing the pathogen on the surface of an inanimated object, And then if anyone else comes later and touches that inanimated object, they could get exposed to that pathogen if they don't wash their hands and now they touch their eyes, mouth, nose, they could get exposed to that pathogen. We call that fomites transmission. And in the case of fomites, examples, well, the SARS coronavirus 2 that is circulating now has been, is a possibility that it could be transmitted through fomites. Influenza, again, can be transmitted through fomites. Tuberculosis, Mycobacterium. Tuberculosis can be transmitted like that too. MRSA, methicillin-resistant Staphylococcus aureus, also transmitted through fomites, a very good example of that transmission. And here we have pathogens that can be transmitted through the oral fecal route. Food, water could be contaminated with these pathogens, and then now a person could ingest that food, that water that is contaminated. Typical examples of these, poliovirus, hepatitis A virus, Giardia, Lamblia. This is a protozoa that produces gastrointestinal infections. Vectors are also considered indirect transmission. And a vector could be, for example, a mosquito, could be a tick, could be a flea, that they now bite a human and they inject the pathogen in that human. Many different examples of diseases that are transmitted by vectors. Dengue, for example, dengue virus, malaria, Plasmodium falciparum, Zika virus, also transmitted through vectors. And these three are all examples of those transmitted by mosquitoes. We will talk in class about other diseases that can be transmitted by ticks and flea. Actually, we just talked about flea, disease transmitted by flea recently, the plague. We will talk more about it in the future. And then finally, we have the droplet nuclei. Droplet nuclei, these are, very tiny droplets that are produced when we cough, when we sneeze, when we talk. Very tiny droplets where the pathogen is suspended in air and can travel more than 1 meter. These small particles, well, we're talking about 1/4 micrometer size. They can remain airborne. They are airborne. They can remain airborne for hours in the air. And then if a person sneezes now and walks away, those particles will remain there suspended. And the next person that maybe walks by in 2 hours could be exposed to that pathogen that was left there in the air, suspended. The typical examples of diseases that are transmitted by droplet nuclei and that are highly transmissible: measles, tuberculosis, TB, by the Mycobacterium tuberculosis. So the bacteria that produces this, Mycobacterium tuberculosis. And varicella zoster virus, that is here in the vertical transmission, well, can be also transmitted through droplet nuclei. Let's now go into an important topic for many of you who are going to be future, nurses or working in the healthcare system. This is the case of healthcare associated infections. we call healthcare-associated infections or nosocomial infections. We're there talking about the same thing. These are diseases that are transmitted to a patient in the context of a healthcare facility. Let's say I go to the hospital for a surgery, and then during my time at the hospital, while I'm recovering, I acquired an infection there. An infection that maybe was acquired, maybe because the nurse who was taking care of me was not taking good precautions to transmit a disease from one patient to another one. Maybe was not changing the gloves, for example. Or maybe, in some of the devices that are used to, like for example, a catheter or a needle, the disease was transmitted to the patient in that way. Maybe during surgery, the room, there was some pathogen in the room that was not eliminated by thoroughly cleaning that before the surgery. These are all ways in which those pathogens could be acquired, at a healthcare facility. Many of these infections, they may result in, presence of bacteria in the blood, bacteremia, infections of the urinary tract, respiratory tract infections, and also infections of that happen at the surgical site. And then 17% are other reasons that could produce those infections. This table outlines some of the most common healthcare associated infections. I will say, among them, I will stress Staphylococcus aureus, Pseudomonas, and Clostridium difficile. We will talk about those in class when we start to revise particular pathogens. We will talk about these healthcare associated infections. Now, towards the end of today's class, I just want to talk about emerging infectious diseases. Emerging infection diseases, these are new infections that we didn't know before and all of a sudden appear. I will say probably the case that you're more familiar with currently is this new virus that jumped to human, the severe acute respiratory syndrome coronavirus 2 that is circulating. But there are many other examples of these new diseases, Ebola, HIV, Middle East respiratory syndrome. This is another coronavirus. Prion diseases, a new recombinant flu virus that may emerge at some point that could be considered an emerging disease. And then we also have the term re-emerging to describe diseases that at some point we thought that we had them under control, but all of a sudden, the cases start to increase again and they re-emerge. And that is the case of malaria and tuberculosis. There are many factors that can contribute to the emergence and re-emergence of these pathogens. One clear factor could be microbial evolution, a pathogen that starts to mutate, that acquire new genes through horizontal gene transfer, and all of a sudden becomes, more virulent or also more resistant to treatment with antibiotic. That could result in the emergence of, a serious new strain of a particular pathogen. Complacency, this is an interesting factor that we could control. Complacency, as an infectious disease is on the way to be controlled. We have that tendency to stop paying attention to them. And when they have not been yet eliminated, we stop thinking about them. Or the governments, for example, they don't continue funding projects that aim to control those diseases. And all of a sudden, they start to re-emerge again. And the number of cases start to increase all of a sudden. This also applies to those of us living in the developed world. Even if we accept that the danger exist, we have this tendency to believe that the real danger is to other people, particularly, poorer people living in distant countries, and we're totally wrong about that. We believe that when we faced a major epidemic, like the one that we are living now, for example, the government and the scientific community will be able to find a solution immediately to protect, us, to protect our families, and that make us, feel safer and less aware of the risk of contracting many of these diseases. Changes in human societies is another factor that is important in the emergence of these diseases. You have a picture here of daycare centers showing you how that, for example, with many infants that maybe don't have the best sanitation and hygiene to interact with each other, many contagious diseases could then spread, especially those pathogens that use the oral fecal route of transmission, like bacteria like Shigella, Salmonella, Giardia, the hepatitis A virus. There are also many other factors that contribute to emergence and re-emergence, and I'm mentioning here, for example, advances in technologies, the use of contact lenses. If the person does not use the proper disinfection techniques, that may facilitate the infections that start in the eye. Population expansion is an important factor. People move to new areas previously inhabited by humans, and they get in contact with reservoirs of new diseases all of a sudden. Another important, well, you have here the, development, mass production and distribution and importation of food, the war and civil unrest that can also, disrupt healthcare infrastructures and disrupt efforts towards the eradication of diseases like distribution of vaccines, and may all of a sudden, start outbreaks in refugee camps with poor conditions, or in a country that is in the middle of a war. Those are, reasons for diseases to emerge or re-emerge. And then also, finally, climate change. Warm temperatures, they favor the reproduction and survival of some vectors that could transmit diseases like, for example, dengue, Zika, or Lyme disease. And all of a sudden, because of changes in the environment, in the climate changes, places that may get warmer now welcome those vectors to those areas where they were not present before. And now those diseases start to show up. This is the end of today's lecture, and our next lecture will be about antimicrobial agents.